Hey, how's it going? Today we're going to be talking about the origin of life. So this is kind of to wrap up our evolution unit and to kind of look at the diversity of life and how life all started. So the origin of life is started by just the right combination of events and chemical processes. In some ways, some people say it even happened by chance that something so complex could happen. When we're looking at the evolution of life, we look at a lot of things. So we would look at things such as a cladogram, which we worked on last week. And we see that we are most related to fungi, actually, but that there are other things in life, such as bacteria, archaeobacteria, protists, and plants. We can also look at the timeline of all of life, starting with the formation of when Earth started to when plants and animals first showed up. So that has to do with evidence, but now let's look at some other parts of how life started. So in order to see how life started, we have to talk about what is life. So you might remember from freshman biology, we looked at a couple of characteristics that make up life. So those characteristics have to do that we have cells. So all living things are made of cells. They respond to stimuli. So this was especially true with the plant project that you did. They responded to different types of light. They responded to different responses to light. They regulate their internal process, meaning they have homeostasis. They can regulate themselves compared to the environment. And some examples of that could be sweating and shivering in people, and homeostasis means you're maintaining your balance. So a lot of times in my room it's way too cold, so you can keep yourself warm, and that's part of homeostasis. You also use metabolism, such as energy, to grow and develop. So metabolism is actually something we're going to hit on next. So all that stuff about photosynthesis and cellular respiration is part of being alive. You develop, so you change and grow over your lifetime, and that you reproduce. So last week when we talked about bacteria and viruses, they're able to have DNA and RNA, and that because they're able to reproduce, they're able to adapt and evolve over time as a species. So there's a couple different hypotheses or ideas behind how life started. And there's always the idea of special creation, so divine intervention, the grand creator. Remember that we have a hypothesis, we have speculation, an idea, but the issue with this is it's not testable. We can't go back in time and observe a special creator. There's also the idea that maybe living material came from off of this planet, and that one is starting to gain more and more ideas. There have been some issues with contamination that people critique this, but the idea that maybe outside of Earth life came, and then that's something that we can test because we can look for things like proteins and carbon structures on other planets or even meteorites to see what origin it came from. The other idea is spontaneous abiotic origin. So this is the most common hypothesis that most people bring up, is that life evolves spontaneously. So because of things like lightning and just the right combination of carbon molecules occurring in the right location, we can then bring life about. So what were the conditions on early Earth that might have brought about this third hypothesis? We're looking at an atmosphere that was reducing. We have water, carbon dioxide, nitrogen, but lots of hydrogen and a lot of electrons. There was no oxygen at this point because we did not have photosynthesis. And once we had photosynthesis and life starting, that changed the composition of the atmosphere. But at this point, there really wasn't any to affect the Earth. From energy, so what could have caused this? Lightning, UV radiation, and there was some volcanic activity on the Earth at this point. So what's missing from this that was able to give us life? We didn't have organic molecules that broke down quickly. So today we have a lot of oxygen, and you can consider those oxidizing agents. So I believe one of our students brought up the idea that we're all slowly dying, and it's just that oxygen is breaking us down. And that's the idea behind this argument, that there wasn't a lot of oxygen, so things aren't breaking down. So where could these molecules have come from? There's a famous experiment where they were able to create these molecules by putting them in an area that they used to simulate it, and they were able to use lightning simulated by these electrodes in order to create kind of these complex molecules, things like carbohydrates and proteins. So there was a couple of experiments from this, Alperin and Haldane, but the main one is that most people know of is Miller and Urey. So some people even recreate this, and they were able to form amino acids and even adenine. So the main idea behind ATP, and one of the main molecules in DNA just by putting molecules that they thought might have been in this primitive atmosphere together with water and electrodes. They were able to spontaneously form. Even later on, he was able to form hydrocarbons, so CH bonds, fats, lipids, and other things. And by putting those together, it was able to suggest the idea, this is strong evidence, that we might be able to create things this way. So now we have the molecules, but how do we get across making cells? So to get a cell, it's a little bit more complicated. It's not just molecules together, but we were able to come up with this idea that protobionts were the first cells. And these were really simple cells. They didn't have all these complex organelles that you might remember, such as mitochondria. But the idea was you were able to create a bubble, and by being able to separate the inside from the outside, you were able to kind of create the beginnings of a cell membrane, something that you can control from inside and outside. So we've got sort of a cell with a cell membrane. We've got some basic molecules, but how do we get genetics? How do we get natural selection? And that started with RNA. So the differences between RNA and DNA is that RNA is a lot simpler. It's a simple single-strand molecule. And from there, they were actually able to thinking, okay, well, maybe RNA was the first molecule, and from there, we were able to get DNA. You might recall from the past that RNA has multiple functions, especially in the central dogma for tRNA and mRNA. So we now have an idea of heredity, we have an idea of cell membranes, we have an idea of how these molecules started. So now we just want to look at some key events on how this all came about. You might have heard of the clock analogy, where humans have barely been a few seconds on the clock of life if all the history of life was 24 hours. But just to pick on a couple of key events that you might want to remember, life originated 3.5 to 4 billion years ago. From there we had the prokaryotes. So recall, prokaryotes lack nuclei, so these are organisms that occurred first. They don't have a nuclei, so these are things like your bacteria. And we even have fossils of these, which is pretty amazing that they're leaving imprints, and that's how we can time them. Now we have modern bacteria, which you will see in the past lecture where I talked about how there are things like cyanobacteria, which aren't actually bacteria, but other prokaryotes, things like Staphylococcus, Diplo, Streptococcus, which are all forms of bacteria. You might also have heard of stromatolites, which are early bacteria that resemble colonies. So a lot of bacteria actually go in colonies and don't remain by themselves. So here's another kind of diverse origin of life that we have. We find these a lot in kind of areas that are not dominated by other organisms, and they act kind of on the extremes. Now we're starting to get cyanobacteria, and we're starting to get an oxygen atmosphere, and this is through photosynthesis. With the buildup of photosynthesis, we're getting more oxygen in the atmosphere, and that's causing some changes. So when this atmosphere is changing, we now can see that in geology. So you might know from APES or other courses where now we can have different imprints because of oxygen in the atmosphere that weren't possible before. So from that we have our first eukaryotes. The idea behind how we got our first eukaryotes is the endosymbiotic theory, that a prokaryotic cell was able to absorb another cell and create these other structures because of infoldings and by absorbing other organisms. And this gave rise to our modern day organelles such as nucleus and endoplasmic reticulum. So the prokaryotic cell is usually seen as the ancestral cell, and the eukaryotic cell our modern day cell. So how do we develop this? We want specialization, increased efficiency. Just like we have organisms that are multicellular with different jobs, the same is true of a eukaryote. You can now specialize and have different jobs. The endoplasmic reticulum is responsible for doing proteins, the nucleus is responsible for storing DNA. This isn't possible in something like a prokaryote without a whole lot of specialization. Again, here's another example of the endosymbiotic theory where you absorb something. Even today, the mitochondria, which are responsible for energy use, have a different DNA pattern than the nucleus, and there are a lot of genes that are only found in your mitochondria. Fun fact is that your mitochondrial DNA is absorbed from your mom, and it's inherited that way through the egg. You don't receive very many, if at all, mitochondrial DNA from your father because of the sperm. And again, here's a summary of what I talked about how we got mitochondria. And again, so this endosymbiotic, endo meaning within, symbiosis is a relationship between the two. So this is also, once we were able to come up with mitochondria, it's the same argument for how we were able to get chloroplasts. So we have something that's photosynthetic, absorbed, not digest, but was able to benefit both the cell and the chloroplast. And we were able to come up with our plant cells this way. So who came up with the theory of endosymbiosis? Lynn Margulis, a pretty famous person in the world of science. She was able to look at multiple forms of evidence, both structural. She saw that there was a lot of bacterial shapes to them. She looked at genetics, so mitochondria and chloroplasts have circular DNA like plasmids, and they're also functional. They're completely independent. Your mitochondria and chloroplasts do not exactly need DNA. Over time, there has been more connection between the two, but mitochondria and chloroplasts can act independently of actions in the nucleus. There was also the Cambrian explosion, so for some of you guys who did the cladogram with the little guys with antennas and plates, this was a huge period of time when there was a huge explosion, or a lot of different animals, quote-unquote animals, diversifying and trying different things. There was a lot of undeveloped niches in this new world, and part of that was the Cambrian explosion, creating a lot of very interesting looking organisms. And those organisms were multicellular eukaryotes. So a lot of animals in our fossil record are occurring within 10 to 20 million years because there isn't any animals. And so here's some of those interesting guys that happened in our Cambrian explosion, trying new things, new body shapes in order to figure out what works best for the environment. So here's a breakdown of some of the other periods in our time, from the Precambrian, Paleozoic, Mesozoic, and Cenozoic, including kind of your dinosaurs and how we've gotten to today. You'll also notice changes in geography, which is related to biogeography, and why we have kind of convergent evolution and ancestors that are really far apart. So just for your reference, we have extinctions, and that happen in different points in time. Extinctions are mass changes where we lose a lot of biodiversity in an area. So there's the Cambrian explosion, where we had a lot of different families, the Permian, and the Cretaceous. So what happened for the Cretaceous? So this is the idea behind why dinosaurs are extinct. We have an impact crater, and that's changing the conditions of the Earth, making it very difficult for photosynthesis and rapidly changing the quality of the air. From there, we were able, because the dinosaurs were no longer around, we had rapid mammal evolution. At 125 million years ago, we had adaptive radiation, where the mammals were being able to fill in these niches that were no longer occupied by the dinosaurs. So this kind of wraps us up for classifying life. When we have all these types of life, we want to classify them into different systems, and this has been an argument and debate over time. So we look at molecular data, and this was the idea behind BLAST and a lot of bioinformatics. We can look at them and figure out who is more closely related to who. And currently, right now, we have a 3-domain system, but even last summer, 2013, I saw that there was some argument on changing these domains. But the most recent edition of our textbook suggests there's bacteria, archaea, and eukaryotes. And these are super kingdoms, and the main idea behind it, we have bacteria, archaea, which are extreme bacteria, and eukaryotes, which is what most people tend to think of when they think of life. Here are kingdoms within that, with the focus on bacteria, extremophiles in extreme environments that are extremely hot, extremely sulfuric, or extremely cold. And here is our eukaryotes, which tend to include protists, animals, plants, and fungi. So if you have any questions, again, please feel free to shoot me questions, calls. I will be happy to take them all. And people always wonder, is there life elsewhere? A question for another topic, another class. There are actually biology classes on astrobiology, on whether there's life on other planets, and whether or not that life is like ours on Earth. So for right now, I believe there's found arsenic-based as opposed to carbon-based. So thank you for your time, and I look forward to seeing you next time. Bye.